Hello everyone. Welcome to Unacademy Future Doctors. Am I audible as well as visible? Let me check. Am I audible? Let me check. Am I audible? Welcome to Unacademy. Here in this channel, we are posting videos for MBBS university exam going students. These videos are free to access and they are very helpful for MBBS university exam going students. So if you have anyone in your family, friends, or juniors, you can share all those videos with them. Okay? Now, I'm myself Dr. Dipti Kariya. I'm MD PhD Physiology. I'm Associate Professor and educating MBBS nursing and physiotherapy students, and I have more than 17 years of teaching experience. Now, let me explain something about our subscription. We have two subscription, one that is plus subscription. Here in this plus subscription, you can choose the best from the best. In this plus subscription, you can access both live as well as recorded classes. You can study on the device of your choice. You can learn from India's top educators for your medical exams. You can access question bank, which is having more than 25,000 questions. You can compete in live tests as well as quizzes, and within 12 months, we are coming with our printed notes. Now, second one, that is iconic subscription. Here, your access to the best from two of the best. And which are these two? One that is Unacademy and Prep Ladder with Unacademy. All the features I have already explained. And second one, that is Unacademy and Prep Ladder with Prep Ladder. The features are you can have clinical and integrated essentials. You can have video lectures from the Dream Team. You can have Question Bank 3 with active guidance, system tags, and more. Rapid revision, snapshots, treasures, and 2021 notes. You can subscribe from your mobile, either Plus or Iconic subscription. You can use my code DIPTI10 and you can get extra 10% discount on each subscription. Longer the duration of subscription, cheaper are the rates. So if you are first or second MBBS student, I prefer that you should go for long duration subscription. Okay. Now, let us start with today's topic. Today we will discuss about cellular immunity, cell mediated immunity. Okay. So in last class we have discussed about humoral immunity. If you want to know about humoral immunity, you can go through my previous class. Okay. Now, so cellular immune response may be asked as a short note. Cellular immunity or cellular immune response. Okay. Now, here, what is the cellular immune response? This is included in acquired immunity. That is acquired, we have discussed. Acquired one, that is also known as specific immunity. Okay? Specific immunity. Here antibodies, or you can say here they are T lymphocytes, not the antibody. Antibodies are in cellular immune response. These T lymphocytes are produced against particular antigen. Therefore, it is known as specific acquired immunity. Okay. So this T cells, effector T cells means those who are working, and macrophages. These two cells are very important for development of cell mediated immunity. Okay? Now, role of cell mediated immunity. Cell mediated immunity protect the individual, or we can say host, against which infection? Our cell mediated immunity protects us against one, that is, you can see viral infection. You can also write down along with me: viral infection, fungal infection, intracellular bacterial. Here this is difference between cellular and humoral immunity. Cellular immunity protects us against intracellular bacterial infection. Whereas here, B cell mediated, antibody mediated, or you can say humoral immunity protects against extracellular bacterial infection. Okay? This is the difference. Why intracellular? Because as all of you know that we have cytotoxic T cells, killer T cells, they destroy the cell. So intracellular bacteria also can be destroyed here. Okay. For example, which are these intracellular bacteria like Mycobacterium tuberculosis, Mycobacterium leprae, brucellosis, Mycobacterium brucella. These all are intracellular bacteria. So against this, our immunity protects us. Okay. Now, next one, that is, it also participates in allograft rejection. Allograft means graft that is given from one individual to another, that is rejected by this immunity. Okay. Also, it participates in delayed type of hypersensitivity reaction. Delayed type of hypersensitivity reaction. And they are also associated with autoimmune diseases. Okay. And provide immunological surveillance. We will discuss what is this immunological surveillance. Okay. And also immunity against cancer is also provided by this. Against cancer is also provided by T lymphocytes. So these are the functions. Suppose if you have been asked that functions of cellular immune response, so they are: number one, protect the host against, you can also write down, viral, fungal, and intracellular bacterial infections like Mycobacterium tuberculosis, Mycobacterium leprae, Mycobacterium brucella. They also participate in allograft rejection associated with a delayed type of hypersensitivity and autoimmune reaction, and they provide immunity against cancer. Okay, now next one. Here also in cellular immunity also we have primary and secondary cellular response. Now, what is this primary response? As we have discussed, primary response that is produced by first contact of antigen. When antigen enters first time, antibody production increases. Here, this one is primary. And here you can see latent period is long. You can see here, this is latent period, means infection, time of infection, and time at which antibody production starts in our body. This is latent period. It is more. Second thing, amount of antibody is less as compared to secondary response. Whereas here you can see in secondary response, latent period is short. Within short period of time, antibody production and you can say T lymphocyte production increases, and this T lymphocyte titer in the plasma is high. Okay. So secondary response, as we all know, second exposure to the same antigen. Suppose we have infection with some virus. Nowadays COVID is common, so COVID infection. Okay. So antibody production or T lymphocyte production in the first response is less. The second time when the same virus attacks, then large amount of antibodies and T lymphocytes are produced. Okay. Now, stages of cellular immune response. Okay. If your question is cellular immune response, first of all, you have to write down it is because of T lymphocytes, role of it, okay, and primary, secondary response, then stages. So here, what happens first? You can see first step, that is antigen processing and antigen presentation. You can see here when antigen enters in our body, first of all, it is phagocytosed by macrophages and other phagocytic cells. Okay? You can see here this is macrophage. Macrophages cause phagocytosis, and this product of phagocytosis, they are displayed on the receptors on the macrophage. Here it is MHC2. this fragments, this is antigen, okay, yellow colored one. These are fragments of antigen. After destruction, they are displayed here. You can see here. And this is displayed to the lymphocyte. Okay, so therefore, these macrophages are known as antigen presenting cells. Okay, so first step is over here. Second step, that is recognition of antigen by lymphocytes. These T lymphocytes, they have antigen recognition receptors. Other name for this antigen recognition receptor, that is specific surface receptors. What is their function? That is to recognize the antigen and interact with single antigenic determinant. Okay? Then here, these T lymphocytes, they are mainly of two types. One, that is CD4 plus cells, and second, that is CD8 plus cells. Why the CD4 and CD8? Because these are the receptors. They're present on that particular T lymphocyte. So it is known as CD4 plus and CD8 plus. Yesterday also we have discussed CD8 plus cells. They recognize combination of foreign antigen with MHC1. I told you, one is like O and E. So here this is 8 like this, or cytotoxic T. So we have discussed yesterday also. Suppose you can see here, this is your antigen presenting cell. Okay, antigen presenting cell has MHC1 and 2. We have discussed MHC1 is present on all the cells, whereas MHC2 are present on the B lymphocytes and macrophage. Okay, so MHC1 is 1 and 2 both are present here if it is macrophage. Okay, so now your antigenic products, they are present on MHC1 and MHC2. When they are present on MHC1, they present this product to this cell, CD8 cell. And these products which are present on MHC2, they are presented to CD4 cell. Are clear? So CD4 cells, they recognize combination of foreign antigen with MHC class 2. So this is your second step. Okay. MHC2 present foreign antigen, as I told you yesterday, to helper cells like this H. So easily you can remember MHC2, and then from 2 you can make H. Okay. So H, helper. Other name for helper cells, that is CD4 plus cell. Okay. Now activation, third step. Activation, or other name for it, that is T lymphocyte differentiation. Okay. Here, this CD8 plus cells, when they proliferate, they form two types of cells. Here in this diagram also you can see that CD8 cells, they form suppressor and cytotoxic T cells. Okay. How can they be differentiated? When they come to know that MHC1 has antigenic product. So binding of MHC1 with antigen, it activates CD8 cells, 8 plus. Okay, and they are differentiated into cytotoxic and suppressor T cells. Okay. Then comes next CD4 plus cells. This you can see here. These are CD4 plus cells. This one. CD4 plus cells. These are also known as helper T cells. Here, I have forgotten to tell you this thing. CD4 and CD8, they are nothing but cluster of differentiation proteins. These are glycoproteins. You can see here, suppose you have this T lymphocyte. Okay? On the surface of T lymphocyte, we have glycoprotein. Name of this glycoprotein, if this is helper, then this glycoprotein is CD4 plus. If it is suppressor or cytotoxic T cells, then the glycoprotein is CD8 plus. Okay, so this CD4 type of cells. These are helper T cells. They have CD4 cluster of differentiation. Okay? Now, the CD4, they are activated when the foreign antigen, you can see here, foreign antigen which is present on your MHC2. Okay? As I told you, easily you can remember, and this thing you try to remember so you can't get confused. Again, MHC1, 1 है तो हम 1 लिखते हैं here. तो कौन से सेल आ गए? साइटोटॉक्सिक and suppressor T cells. This 1 and this C, like this you can write, suppressor T. Okay, and 1, these two are known as CD8 plus cells. And MHC2, अगर MHC2 के ऊपर एंटीजन है तो then it stimulates your, यहाँ पे हम 2 लिख रहे हैं पहले, तो उसको बना दो H, helper T cell. Okay, and helper T cells का दूसरा नाम है CD4 plus cells. Okay, so you can see here, these are helper T cells which are stimulated. How are they stimulated? When MHC2 with antigen gets presented to them. Okay, and they are responsible for delayed type of hypersensitivity. They are responsible for stimulation of helper T cells, and this is known as TT cooperation. Means when T cells are activated, now they differentiate different T cells. Means from your helper T cells, you can produce cytotoxic, suppressor, and memory T cells. So this is TT cooperation. Okay? Now, next is memory cells. This memory cells, T lymphocytes, you can see here, these are memory T cells. This one. This memory T cells, they spread throughout the lymphoid tissue in our body. Okay? And whenever there is subsequent exposure to the same antigen, then this memory T cells, they are helpful. And they release or they produce T cell at a faster rate than fast response. So this is more powerful than fast response. This is the role of memory T. Now, next very important thing, mechanism of action of, you can say that mechanism of action of T cells. Main mechanism, how they destroy. This must be there in your answer. How they destroy this cell which is infected with virus or bacteria or anything. Okay, so you can see here, this is your virus infected cell. Here there is the role of your cytotoxic T cells, other name is CD8 plus cells. Now, here what happens? The cytotoxic T cells and other variety of cells, natural killer cells, these two, they are responsible for this action that is also known as attack phase. Now, T cells are ready to attack. Okay, so this T cell, cytotoxic T cells, they have receptor proteins. You can see here, they have receptor proteins. So this receptors, through this receptors, they bind with this cell, which is the cell which is infected with antigen, whether it is virus or bacteria or anything. Okay, so what happens when this binds with your T cytotoxic cell binds with the virus infected cell? Then next thing is there are porphyrin. Porphyrin other name is their hole forming agent. Means like punching machine our punch karte na. So they make holes here in this cell. Virus infected. Okay, so these are porphyrin. And through this hole, different enzymes, different hydrolytic enzymes, different lysosomal enzymes, they get enter inside the membrane, inside the cell through punching or through making holes. So these are porphyrin mediated killing. Okay, this is lysis through cytotoxic substances. Okay, now there is also apoptosis. Apoptosis is also known as programmed cell death. Programmed cell death. And for programmed cell death, these cells, they also secrete tumor necrosis factor B. And this tumor necrosis factor beta, how do they work? They increase calcium permeability. Okay, permeability of this cell. And when calcium enters inside the cell, that causes destruction of nucleus, and after nucleus is destroyed, there is programmed cell death. So this is the mechanism of action of cellular immunity. This must be there in your answer. Okay. Now, next is helper cells. These are highest in number, most numerous from all the cells. As we all know, four varieties of cells are there: T helper cells, T cytotoxic, T suppressor, and T memory cell. Okay. So your helper cells are most numerous. You can say they are 3/4 of all the cell. Mainly there are two types, Th1 and Th2. This Th1, they secrete, you can see these are Th1. So they secrete interleukin 2. Here, they secrete interleukin. You can write down also interleukin 2, gamma interferon. Yesterday we have discussed, you can go through that video, gamma interferon, how they work in viral infection. Also, tumor necrosis factor beta. They work by secreting all this. Okay? And Th2 secrete interleukin 4, 5, 6. And here they stimulate B lymphocytes also. Okay? So this are important for antibody mediated immunity also. So you can see here T cells are also important for cellular as well as antibody mediated immunity. These T cells are targeted by human immunodeficiency virus. So what happens? Both the varieties of immunity are lost here. Okay? Now, next is suppressor T cells. Suppressors are also known as brake. So they are regulatory T cells. What happens if there is no brake? Then there is accident. So here, if suppressor cells are absent, then cytotoxic cells, they cause destruction of large amount of cells. Sometimes body's own tissue is also destroyed. Body's own tissue is destroyed. So their activity must be regulated by cytotoxic, by regulatory or suppressor T cells. Okay. So this we have discussed all about which immunity? Cellular immunity. Now, next topic here. This is immunological tolerance. Now, what is this immunological tolerance? We will discuss. This will be asked as like what are the abnormalities of immunity? Your question, we will discuss questions in next class, which questions will be asked in your topic, from your topic. Okay, so immunological tolerance means what? We can tolerate antigen, means our body is not responsive, or you can say unresponsiveness to, you can write down this, unresponsiveness to antigen, that is immunological tolerance. Okay, now there are two types of tolerance. Number one, that is natural tolerance, means we are unresponsiveness to our self antigen. This is natural tolerance. Our body will not produce antibodies or T lymphocytes against our own antigens. Okay, now so this natural tolerance, it develops in the intrauterine life, just before birth or sometimes after birth also this is been developed. Okay? Now, so you can say that all the antigens, suppose in intrauterine life, when the fetus is exposed to any antigen, okay, and during this period natural tolerance is been developed. So this immunological immaturity stage, suppose if we are stimulating the fetus with strong antigen, then also the fetus will stop producing antibody against that antigen. This is natural tolerance, naturally not trying to produce. Okay? And here at this time, fetus is trying to know, or the baby is trying to know the difference between self and non-self antigen. Okay, so what happens here? Some clones of the T lymphocytes are produced, but they are destroyed. Okay. Second thing is acquired tolerance. Acquired means it is developed after birth. Okay. Now, what is the mechanism of tolerance? You can see here there are three mechanisms we will discuss. One is clonal deletion, second is clonal energy, and third is suppression. Now, what is clonal deletion? Here, what happens? There is also known as negative selection. This one is clonal deletion. Deletion means cell death. So you can see here, there are receptors present on the B and T lymphocytes. They recognize antigen. Many of this non-self T lymphocytes, they are eliminated if they are there in this early fetal life. So they are, this clone is produced against self antigen, clone of T lymphocyte, but it is destroyed. Okay, so you can say selectively it is deleted or eliminated. So this is clonal deletion, first thing. Second thing is clonal energy or immunological silence. Here you can see this is energy. It is not been deleted, but it is unresponsive. These clones are produced. Clone means asexual development of cells, cell growth from one cell. This is a clone. Okay. So this clone is produced. It will not be removed, but it remains silent or it is not reactive. So it will not produce immune response. Okay. Then third, that is suppression. Here, these clones, already they are present clone of B and T lymphocytes. They recognize the antigen, recognize self antigen or other antigen, but that is suppressed actively. Our body tries to suppress it actively. Suppressed. This is third mechanism. Okay, so these are three mechanisms. Now another is tolerance to fetus. What is this? Because fetus is genetically different than mother, but immune response cannot be produced in the mother. This is one of the very good example of immunological tolerance. Now, which are the factors that prevent this immunological tolerance? One is placenta. Placenta is having tropoblastic cells. Now, these tropoblastic cells, they lost their immunogenic capacity, okay, because they have decreased MHC antigen. So, no immunogenetic activity they have. Second thing, there are mucoproteins. They coat on the cell surface. Okay? Suppose if some cell is there which is acting as foreign body, but some mucoproteins are there, they cover them. So they are not becoming antigen. Third, this anti-MHC antibody, suppose they are produced in fetus, they can this which are produced in mother, they cannot enter in the feet. That is prevented by placenta. Okay. Next is alpha fetoprotein. This is also acting as immunosuppressant. Alpha fetoprotein is also produced. Progesterone level is high during pregnancy, which is also immunosuppressant. Fetal T cells are also there. So this, what is the role of fetal T cells? This fetal T cells, they are activated when fetus is exposed to, this is fetus, when fetus is exposed to mother's T cells. Okay. Okay. And suppose mother's T cells, they enter in the fetus. But what the fetus do is, fetus suppresses mother's T cells. So fetal T cells suppress mother T cells. So these are all the mechanisms to prevent immunology or immune reaction between mother and fetus. So this is all about today's topic. Here from today's topic, the question may be asked, that is define immunity, mention its types, and discuss cell mediated immunity. That is long question may be asked. This is important, means it is asked 3-4 times in university exam. Short questions from today's topic, that is what is immunity, describe cell mediated immunity, and other question is T lymphocyte. So this is all about today's class. Thank you. If you like my video, Please press the like button, you can share and you can subscribe our channel. Thank you.